Let's start today's class. So last day topic fungal completed topic cell wall structure, right? Fungal cell wall structure and fungal cell wall structure composed of chitin and some proteins are also present along with the chitin, manoprotein, glucoprotein, etc. Am I right or wrong? Cell wall structure of fungal cell wall structure. লাস্ট ডে কমপ্লিট করেছি তো সেল ওয়াল হ্যাঁ সেল স্ট্রাকচারের মধ্যে সেল ওয়াল কমপ্লিট করেছি তো ওকে সো রিপ্রোডাকশন reproduction in fungi. Fungal reproduction can be done by either asexual or sexual. Asexual reproduction mode or it could be sexual reproduction mode. Both possible. So start with asexual mode of reproduction in fungi. Various way by which fungi can do asexual reproduction. First asexual mode of reproduction style is fragmentation. Fragmentation. Look at the word fragmentation. What does it mean? Fragmentation means fragment of a body. Body is so the full body structure mycelia or hyphae become fragmented into pieces and each piece has the capacity to regenerate a full whole structure of mycelia or hyphae. That's called fragmentation. বোঝা গেল আমি যেটা বলতে চেয়েছি? What is fragmentation? So fragmentation means in this process hyphae get broken into fragments. And each fragment has the capacity to develop new hyphae. Just look at the figure, fragmentation style. This is a hyphae structure, a hypha of a mycelium, correct? First of all, this hyphae is broken down into pieces from that part, from that part. So here I break down this hyphae into three parts. Two position থেকে break down হয়েছে, three parts এ break down করবে. So after that, the broken pieces are this one piece, second piece, third piece. Now কি হবে এই পিস গুলো কি জয়েন করে নিউ হাইফি তৈরি হবে না each of the pieces form like the parent hyphae. Clearly I mentioned each of the piece has the regeneration power to make a new parental type hyphae. তার মানে এই পিসটাও this স্ট্রাকচার তৈরি করবে এটাও and third piece যেটা small piece এটাও তৈরি করতে পারে. So after that three hyphae are generated. Three parental hyphae are generated from this part, and from this part. And the structure will be same as the parental. Same as the parental. This is called fragmentation. The parental body break into pieces and each piece has the power to regenerate the parent hyphae. This is the regeneration and this is done by some fungi like Rhizopus, Mucor, etc. Fragmentation done by Rhizopus, Mucor. Fragmentation is the first asexual mode of reproduction style in fungi. Second mode, mode number 2, fission. Very popular in bacteria also, bacteria binary fission. Here also in fungal case, fission can be done. Binary fission is possible. And this is done by yeast, actually show by yeast. Okay, fission means transverse splitting of parental cell into two daughter cell. Transverse splitting of parental cell into two daughter cell, that is fission. And fission is shown by fission yeast. Example. Fission is scientific name Saccharomyces pombe. Cerevisiana. Cerevisi show করে হচ্ছে budding. Saccharomyces pombe. S. pombe. Saccharomyces pombe is the fission type of yeast. Yeast is unicellular organism. That's why fission is most popular in yeast. Fission yeast. Okay. Figure, diagram, fission, process fission. Parental organism yeast. This is nucleus. It is in the stage of interphase. Interphase. Now, interphase yeast move into stage number 2. Early mitosis. The cell grow from the nucleus. Actin ring is formed. From the nucleus, actin ring develops. This is the actin ring, and it split the nucleus into two halves. This actin ring is the red color line. The red color line here is the actin ring. When actin ring is formed, that means the cell is now in early mitosis, not in interphase. Interphase কোনো actin ring তৈরি হয় না. Correct. So now the cell stage here is early mitosis. Followed by the cell now enter into the anaphase. Anaphase means anaphase কি হয় cell chromosome পোলের দিকে ওরিয়েন্টেড হয়, দ্যাট মিনস সেপারেশন অফ ক্রোমোজোম, সেপারেশন অফ ক্রোমাটিড হতে পারে, সেপারেশন অফ ক্রোমোজোম হতে পারে, মিওসিসে ক্রোমোজোম সেপারেশন হয় ফলোড বাই ক্রোমাটিড, এন্ড নর্মাল মাইটোসিসে হচ্ছে তোমার ক্রোমাটিডের সেপারেশন। সো এখানে কি হবে, নিউক্লিয়ার সেপারেট করে যাবে এন্ড ইচ মুভ ইনটু দ্য নিউ নিউ সেল অফ দ্য ইচ পোল, পোলের দিকে মুভ করবে। So after early mitosis, now the cell enter in anaphase, and during anaphase stage, the nucleus splitted and move into each pole like that. Still the actin ring are present. Actin ring actually guiding where the septum is occur. এরপরে কি হবে, নেক্সট স্টেজ septum formation means cytoplasmic division. So actin ring still present and it guided where the septum is formed. Otherwise কি হতো, সেপ্টাম যে কোনো জায়গা থেকে ফর্ম হয়ে যেত, তো কি হতো, দুটো সেল যে ইকুয়াল ডিভিশন হচ্ছে, ইকুয়াল ডিভিশনটা হতো না। Point clear? So now here the anaphase occur. Anaphase occur. Nuclear division, nuclear separation into pole. Followed by, now septum is formed. Septum is formed and actin ring condensed. Septum ring. Green color line is the septum ring. Septum ring formed and actin ring. The red color line is now contract, pull out, because of the function done. Actin ring function done. Actin ring function in karyokinesis, not in the cytokinesis. So actin ring contraction and septation occur. This is the very important step. After that, the cell will be divided. So this stage is called septation. Uh, septation, that means septum formation. Actin ring contraction. Actin ring contraction. Actin ring contract করেছে তো? Very clearly here shown that actin ring now contracted. That means size a small হয়ে গেছে. Followed by the final step, creation of two new daughter cells. Septation occur and two new daughter cells are formed. So that is done in fissionist Saccharomyces pombe. Saccharomyces pombe show fission process for asexual reproduction style. Cell separation. Fission process clear everyone, online students. Diagram clear.
Yes, sir.
Yes, sir.
So mode number 2 is done, fission. Third mode, budding. Mode number 3 is budding. It is also done by yeast, but the species is different. The species is more common, Saccharomyces cerevisiae, not the Pombe. Difference there. S. Pombe versus S. cerevisiae. ডিফারেন্সটা রিপ্রোডাকশন স্টাইল দিয়ে আসবে। তুমি মাথা খুঁজেও পাবে না এস পম্বে এস সেরিভিসির মধ্যে কি ডিফারেন্স। ডিফারেন্স এইটা দিয়ে আসবে, বার্ডিং এন্ড ফিশন। Okay, so budding process. During budding process, a simple outgrowth comes out from the parent cell. Comes out from the parent cell. Then this outgrowth later grow later grows and pinches off from the parent to form independent daughter cell. From parent cell to generate new daughter cell. Like that. Oh, budding process example, Saccharomyces cerevisiae. Process of budding. Then parent is, then an outgrowth is formed, developed. Like that. A extending part. Outgrowth means a extending part from the body. That is the extending part. And the nucleus also divide and migrate to this outgrowth. After outgrowth is formed, the extension of the body formed, the nucleus also extending and migrate towards the outgrowth site. Next, outgrowth, this outgrowth or extending part continuously grow and septation properly done. Nucleus are migrated into the outgrowth. After that, the pinches off from this parent cell. Pinches off মানে বুঝতে পারছো কি বলতে চাইছে? Pinches off মানে হচ্ছে না মানে হচ্ছে রুটি ময়দার লেচি কাটা দেখেছো? জাস্ট এরকম কেটে বেরিয়ে আসবে। That means parent cell remains same, but an outgrowth is formed. Pinches off. This is the new daughter yeast. Developing bird. Parental yeast cell. Budding process clear?
Yes, sir.
Okay. Next, asexual mode reproduction, more popular than other three that I described here. The fourth method is more popular in asexual mode, that is spore formation, or you can say about reproduction by spore. Reproduction by spores. In most of the fungi, it is the popular method, spore form and germinate. Most common method. And there are many type of asexual spore can be formed. Some of the example, asexual spores example. Number one, arthrospore, and another name is oidea. Arthrospore or oidea, it is actually the breakup of hyphae. Arthrospores are produced or oidea are produced after breakup of hyphae. These are formed when hyphae breaks up into small fragments. Check the odia formation in diagram. See, that is a parental hyphae structure, and hyphae breaks down into small fragments like a spore. So this is the odia or arthrospore, asexual mode. হুম বুঝলাম না। ও আচ্ছা আচ্ছা, হ্যাঁ hyphae break down করছে into small fragments, and that small fragment is actually the spore called arthrospore. Type number 2, Conidiospore. Conidia. It is also produced, these spores produced from tip of the hyphae, tips of the hyphae, and these spores do not covered in a sac, do not enclosed in sac. So these spores are exogenous spore, exogenous spore, and also non-motile spore. Now let's look the conidia formation. Fungal hyphae. From that hyphae, the tips form some spore. এতগুলো না, just single, like that. So difference দেখতে পাচ্ছ ওইডিয়া and conidia, difference দেয়। আমি এই জন্য পাশাপাশি ডায়াগ্রামটা রেখেছি। What is the difference between odia and conidia? In first case, odia, the hyphal segmentation or breakdown of it. Hyphae breakdown করে গেছে and breakdown fragments called the spore. এখানে hyphae breakdown করেছে। Hyphae breakdown করেছে conidia? No. Conidia from the tip, this cell, this hyphal cell, the tip cell produces these spores. Hyphae is not breakdown. Hyphae never broken down into pieces in conidiospore. The tip of the hyphae produces the spore, and these spores are open. These spores are not enclosed in a sac. এখানেও কোনো sac ছিল না, এখানেও কোনো sac নেই, but major difference is broken down or not. Hyphal broken down or not. Hyphal not broken down in case of conidia formation, conidiospore formation, but arthrospore, during that the hyphae breaks down into small pieces.
স্যার স্যাকটা একবার বোঝাবেন, এনক্লোজড না স্যাকটা?
স্যাক আসবে, এটা তো স্যাক নেই, এরপরটা স্যাক আসবে, তখন দেখে নাও ডায়াগ্রাম। These two clear? Conidia, odia এর মধ্যে major difference, oidia and conidia?
ইয়েস স্যার।
Type number 3. Sporangiospore. Here, the spores are developed within a sac. or vessel called sporangium at the hypertip. So when spores are covered with the vessel or sac-like structure, the spores are endogenous or exogenous? Endogenous. আগেরটা কি ছিল? Exogenous. So here in this case, the spores are endogenous because as it is covered. Sporangiospore can be motile, usually motile, but sometimes non-motile also. But আগের স্পোরটা ছিল হচ্ছে completely non-motile. Motile or non-motile. Motile and non-motile, how does this motility depends? What structure actually creates this motility? না
flagella.
motility করবে তো flagella. That means motile spores contain flagella and non-motile spore do not have flagella in their structure. So flagella containing motile spores called zoospore. Called zoospores. So zoospore actually part of sporangiospore, which spores have flagella in their structure, which can motile, have the motility. They are called zoospore. And the non-motile sporangiospores that do not have any flagella, and they are called aplanospore. Aplansospore. So sporangiospore can be of two types based on motility, present or not. Aplansospore and zoospore. Zoospore ওয়ার্ড শুনেই মনে হচ্ছে এটা মোটাইল হবে, অ্যানিমেল ওয়ার্ডটা আসছে মানে মোটিলিটি আসবে, দ্যাটস ওয়াই zoospores term are given. So motile spores are called zoospores and non-motile are called aplansospore. And another features, they contain sac, they are endogenous. Now let's look at the structure of sporangiospore. It is also produced at the tip of the hyphae, but the spores are enclosed. The blue dots are spores. It covered in a sac. This sac is called sporangium. এই সেকটা বাস করলে বেরিয়ে আসবে sporangium. The sac is called sporangium, and blue ones are called sporangiospore. Sporangiospore clear? So sporangiospore versus odia or conidia যে কোনো কিছু দিতে পারে, ডিফারেন্স কিন্তু ভালোই আছে, দুটো কি তিনটা পয়েন্ট চলে যাচ্ছে। No sac, endogenous, exogenous, so চলেই আসছে, দুটো পয়েন্ট তো কমপ্লিট। ও ইয়েস, মোটিলিটি দিয়ে আসবে। Both can be possible in sporangiospore, they are non-motile. Clear my point?
Sir, previous ones
Ah,
are non-motile.
please repeat.
Sir, before sporangiosspores, the two spores are
Ah,
non-motile.
these two, Odia
Huh.
and Conidia. Ah, non-motile.
Okay, sir.
Okay. The fourth one, fourth type, asexual spore, blastospore. It is done. Blastospore, the special feature is they are produced from the vegetative cell by budding. So blastospores are developed by special phenomena means budding. এদের ডেভেলপমেন্ট মাইটোসিসের থ্রুতে হয় বাট এদের ডেভেলপমেন্ট ব্লাস্টোস্পোর ডেভেলপমেন্ট ডান বাই বার্ডিং প্রসেস দ্যাটস ওয়াই দিস স্পোর্স আর different from other three. So blastospore. The outgrowth is occurred from the vegetative cell, and from this outgrowth, the small spores are formed. These are spore, blastospore. And this is the outgrowth, means bud formation. Bud. So as these spores are developed from the bud, means budding process, that's why these spores are different from other three and called blastospore. These are common types of asexual spore: Odia, Conidia, Sporangiospore, and Blastospore. All of you clear? Asexual mode, spore formation by through spore formation, and most common method. Answer me. Is it clear to all? Asexual
Yes,
mode,
sir.
the four types of spores in asexual model, spore model, asexual spore formation model. Done. Sexual reproduction, asexual complete. Reproduction mode number two, sexual. Sexual reproduction process occur in 3 stages. Stage 1, stage 2 and stage 3. The stage 1 is called plasmogamy. What is the process? Plasmogamy. Stage number 2 in the sexual reproduction of fungi is karyogamy. And the third stage is meiosis. Plasmogamy meaning fusion of two protoplasts. Coming from two different cells. So plasmogamy word meaning fusion of protoplast means cytoplasmic content all over full cytoplasmic content and fusion. That's called plasmogamy. So after plasmogamy, two different nucleus are close each other. Yes or no?
Yes.
প্লাজমোগামি হলে নিউক্লিয়াস দুটো কাছে আসবে তো সো দুটো নিউক্লিয়াস যখন ক্লোজ টু ইচ আদার ওরা কি বসে থাকবে না ওরাও ফিউজ করবে and the process is called karyogamy. So karyogamy meaning fusion of
Two nucleus.
yes two haploid nucleus. Two haploid nuclei to generate diploid zygote and this diploid zygote enter into the process of meiosis, meiotic event. Zygote, diploid zygote enter meiosis to develop diploid or haploid?
Haploid.
To develop haploid structure or diploid structure after meiosis?
Haploid.
Haploid. Haploid. Okay, how many cells are produced after meiosis?
Four.
One convert into four. Mitosis এ 2 হয় but meiosis 2 events, meiosis 1 and meiosis 2. After that 4 haploid cells are produced. Here not cells, here spores are produced. So zygote enter into the meiosis to develop 4 haploid spores. So here also the final outcome is production of spores. So spores is very common, most popular method, common to develop. But some spores are asexually made that I previously shown. Different types of asexual, but these spores produced by sexual event. So these spores are called another term meiospore, called meiospores. As it go through the meiosis to develop the spores, that's why the spores that develop after these sexual event are called meiospores. আর প্রিভিয়াস যে স্পোর গুলো দেখালাম গুলো কি বলে মাইটোস্পোর মাইটোস্পোর্স the previous spores example sporangiosspore conidiospore, etc. Those are blastospore, those are mitospore. But after the sexual event, the spore that is developed is called meiospore. So spore structure is very common to the fungal. Spores can be from asexual. Spores can be from sexual event after sexual event also. The spores that produce asexual event, mitospore. The spores that produce after meiosis, called meiospores. Clear? So stage 1 is fusion of protoplast. After protoplast fusion, two nucleus are coming each other. So nucleus also will fused each other now. And now this event is called karyogamy. After fusion, two haploid nucleus produce diploid zygote. This diploid zygote enter into meiosis and develop four haploid spore. This event also called, we can also say zygotic meiosis. Zygotic meiosis. Because zygote enter into the directly meiosis event. That's why this event specifically named zygotic meiosis to develop four haploid spores. Now now the complexity is coming. Plasmogamy, that event, the fusion of protoplasm, the stage 1 event. Is it very simple or complex? Complex. And it is complex and it is done by variety of way. The plasmogamy events can be further grouped or subdivided. This event, fusion of two protoplasts. It can be done by plasmogamy event can be done by the stage 1 event, the primary event, planogametic copulation, plasmogamy event can be done by plasmogamy. Oh, sorry, plasmogamy, so that's whole event. Planogametic copulation, after that, gametangial contact, gametangial contact. Third method, gametangial copulation. Fourth method, spermatization. And fifth method, somatogamy. So now we describe each of the event of plasmogamy. So plasmogamy is the complex step, the primary step, the stage number 1 of the sexual reproduction of fungi, and this stage can be done by any of the following ways. Okay, planogametic copulation, gametangial contact, gametangial copulation, spermatization, and somatogamy. Different species of fungi show different process, different event to plasmogamy process. So one by one start করো. Planogametic copulation first. Under plasmogamy, planogametic copulation. Look at the word first, planogametic means here clearly planogametes are formed. Plant তো আসবে না, এখানে fungal cell আসবে, but fungal cell can form gametes. They are called planogametes. So planogametic copulation, the first word meaning planogamete formation. Then the second word says about the copulation. Copulation means mating. Copulation means mating or fusion. Clear? And these two gametes, these planogametes, are coming from two different species to mate each other. That's why sexual events are occur here. And exchange of chromosome, genetic exchange also done in sexual reproduction by that means. Because these planogametes, these gametes contain the sexual nucleus, genetic material, and that is exchanged between the two different gametes. So planogametic copulation, its simple meaning, it involves fusion of two compatible mobile gametes or planogametes. So those fungi that show planogametic copulation produce mobile gametes. Produce mobile gametes. And if two mobile gametes are compatible to each other, they start to fuse and form plasmogamy events completed. And followed by karyogamy and meiosis to develop more. These mobile gametes, this planogametic copulation, in planogametic copulation, three types of gametes can be formed. Three types of gametes are formed. Isogametes, anisogametes, and oogametes. The process by isogamete copulation is called isogamy. Process by anisogamete formation is called anisogamy. And it is called oogamy. What are the difference between these three gametes? In isogametes, the gametes are morphologically similar. Gametes are morphologically similar, but physiologically different. Look at this statement. Morphologically similar. That means the structure, overall structure looks like same. Size, shape, that is same. But physiological, that means internal matter. Is this the same? No. The internal matters are different. That means clearly these gametes are coming from two different fungal species. Then anisogamous. Anisogamous in anisogamy produce anisogametes. Anisogametes, morphological and physiological, both are different. Morphologically and physiologically different. And oogamous, it is higher type of planogametic copulation. Higher fungi they are showing. And here, female gamete is non-motile, called egg, and male gamete, small and motile, called sperm. So here is the concept in oogamy that show by the higher fungal species. Here is the concept is egg and sperm. Means egg are large, non-motile, sperms are small and motile in oogamy process. Now look at the diagram and identify which of the process here is shown. কোনটা কি process? The
Sir,
first
first
one.
one is isogamous, second one is anisogamous, and third one is oogamous.
Correct. Clearly mentioned in the diagram also. Isogamous means fusion of two morphologically similar. দেখতে পাচ্ছ? Same morphologically, but physiologically different. Here it is black, here it is blue. And I mentioned plus minus means they are not same. Plus plus they are compatible or incompatible? যদি আমি প্লাস দিতাম, যদি এটা প্লাস আসতো না, এটা কম্প্যাটেবল ফিউশন হতো না, মাইনাস বলে ফিউশন হয়েছে. So the first process is isogamous. Anisogamous, they are morphologically different as well as physiologically different because one is black and other is coming from different species. That means blue. Anisogamous. And third one is here is the concept of egg and sperm because the egg is non-motile. In previous scenario, both cells are both gametes are motile. Check karo. But here the large cell, the large gamete is non-motile and called gametes. These gametes are called egg or female gamete, and the small cells, small gametes, that is motility, called sperm. So in the oogamous, there is a concept of egg and sperm. But in previous two cases, no egg, no sperm. The gametes are morphologically can be different, can be different and physiological both different. That is in case of anisogamous and isogamous, both morphologically same but physiologically different. Plus minus. Planogametic copulation, clear or not? Everyone understood planogametic copulation process?
Yes, sir.
Yes, sir.
Planogametic copulation done. Next event in plasmogamy, gametangial contact. Gametangial contact. Let's try to explain gametangia. What is gametangia? Gametangia means fungal sex organ, not gamete. So gametangia, here the meaning of gametangia, sex organ. And it is written here contact. That means sex organs now, in that case, now in contact each other between two different species and gametes are delivered. এখানে ডাইরেক্ট গ্যামেট এক্সোজেনাসলি রিলিজ করে না, এখানে গ্যামেটেনজিয়া আসবে, দ্যাট মিন্স কন্ট্যাক্টে আসবে, দ্যাট টাইম ফিউশনটা পসিবল হবে, গ্যামেট ট্রান্সফার হবে। ওকে, গ্যামেটেনজিয়াল কন্ট্যাক্ট, it is between two types of sex organ. It is occur between two types of sex organ, male sex organ and female sex organ. Male sex organ is called antheridium, and female sex organ is called oogonium. So first, antheridium and oogonium contact each other. And male gametes from the antheridium are transferred to the oogonium. By fertilization tube. So first they are contacted each other, oogonium and antheridium, and then male gametes from the antheridium enter to the oogonium by means of fertilization tube. So একটা connection তৈরি হয়, and connection is via fertilization tube, and from that tube the male gametes enter to the oogonium process and done the fusion. Now let's look the diagram. Female sex organ, oogonium. A contact is formed. After that, a fertilization tube formed or occurred. এই যে, হ্যাঁ formed. This is fertilization tube. Contact আসবে, টাচে আসবে, fertilization tube form and male gametes are now transferred to the oogonium. The large structure is oogonium here. Large female sex organ is called oogonium. Male sex organ is called antheridium. কন্ট্যাক্টে আসার পর ফার্টিলাইজেশন টিউব ফর্ম হয়েছে, ফিউশন কি হয়ে গেছে, ফিউজড করেছে কি, ডিড দিস টু সেক্স অর্গানস আর ফিউজড অর নট, নো ফিউশন, only contacted. After that fertilization tube is formed and male gamete is transferred. Clear? And the next process immediately is called gametangial copulation. The process known as gametangial copulation. তো copulation এ কি হবে, এখানে কন্ট্যাক্ট এসছে আর ওখানে কপুলেট মানে একদম তোমার ফিউজড করে যাবে। So a difference that gametangial contact, gametangial copulation. Gametangial contact means they are contacted to each other without fusion. In case of gametangial copulation, they are fused to compatible sex organs. That total sex organ is fused করে। তো sex organ fuse করলে তো gamete fuse করবে। So in gametangial contact, fertilization tube should be formed, otherwise male gamete cannot be transferred. But ওখানে লাগবে fertilization tube, তো পুরো sex organ fuse করে গেছে। In case of copulation, no fertilization tubes are formed. Next process, gametangial copulation. Copulation occur between the entire protoplasmic content of the two gametangia. Gametangia singular, plural gametangia. Gametangia means sex organ, not sex gamete. Clear? Gametes are called planogametes and organs are called gametangia, gametangium. Male gametangium are called antheridium, female are called oogonium. So এখানে just contact ছিল, ওখানে copulate করবে, entire protoplasmic contents are fused. চেক করো, fused each other, all the no fertilization tube, এটা কোনো fertilization tube না, এটা হচ্ছে fusion-এর part, no fertilization tube, that is major difference copulation and contact এ। ওখানে ফার্টিলাইজেশন টিউব হয়েছিল, এখানে পুরো সেক্স অর্গানটাই ফিউজড করে গেছেন, দ্যাট মিন্স দ্য নিউক্লিয়াস অলসো নাও কামস ক্লোজার টু ইচ আদার, all the protoplasmic content are fused. Gametangia, gametangial copulation clear, gametangial copulation clear, gametangial difference between contact clear,
Yes, sir.
spermatization, spermatization, here in this case small male gametes, union of small male gametes or male gametes na, male cell better, male cell or spermatium with female receptive structure. That means one fungal hyphae produce spermatium, small sperm cell, male cell, small male cell, spermatium, and other hyphae produce female receptive, and this union is called spermatization. Look at the diagram. These are not spore, small cell, male cell, spermatiums are produced here in that micelle. And here it is the female hyphal receptive structure. Receptive means that receive the spermatium, যে receive করবে তাকে receptive বলবো, as clear as that. So from that small male cell move into female receptive structure, check কর। The small male cell here from detached and move to the female hyphal receptive structure, and now union is done. This type of union is called spermatization. Sperm cell ke remove করলো, release করলো, these sperm cells are now bind and fuse to the female receptive of next hyphae. Spermatization clear? Followed by next event, last event, plasmogamy, somatogamy. When sex organs are absent, that time plasmogamy takes place by fusion of two somatic hyphae. Of compatible cell. So somatogamy process do not have sex organs. Check karo, sex organs are absent. That time কি হবে? The hyphae are now fused. Structure check karo. Somatogamy. Fungal hyphae. Compatible fungal hyphae. When these two are compatible, these two hyphae very close near to each other and form connection. Hyphal extension, form connection. This is called somatogamy. So somatogamy and gametangial copulation. Most of these students are very confused in the two process. মনে হয় এটা same. No, they are not same. Gametangial copulation, clearly their sex organs are present, antheridium and oogonium. And here there is no sex organ, only the hyphal cell. The extension of the hyphal cells create the connection. So these are different process of plasmogamy. That is stage number 1 means protoplasm fusion occur. Any of this process, protoplasm fusion can be done. It could be gametangial copulation or connection, that depends on fungal species variety.
And it also depends on the conditions.
Conditions based on mainly species, because দেখো কারোর যদি সেক্স অর্গান না থাকে সেটা তো আর কন্ডিশনের উপর ডিপেন্ড করবে না। ওকে সোমাটোগামি ইজ মাস্ট ইভেন্ট, তাই না? আর যে গ্যামেট তৈরি করতে পারে সে সোমাটোগামি কেন করবে? সে করবে হচ্ছে প্ল্যানোগামেটিক কপুলেশন করবে। সো ইট ডিপেন্ডস অন দ্য ফাঙ্গাল ভেজিটেটিভ প্রপার্টি কি তৈরি করছে। ক্লিয়ার? So plasmogamy, different process covered. Next, karyogamy. Simple process after plasmogamy. Stage number 2, karyogamy. Means after protoplasmic content fusion, there is now up to chance to nucleus fusion, because two nucleus, different species of nucleus close each other after protoplasmic fusion. Now they are fused. Already I stated what is the meaning of karyogamy: fusion of two haploid nucleus. to form diploid zygote. And followed by stage number 3, the diploid zygote now enter into the meiosis to form meiospore. Enter into meiosis process to develop meiospores haploid. And there are different examples of meiospores. Example, meiospores or sexual spores. Ascospore, Basidiospore, Zygospore. These are meiospore, not mitospore. তো সেক্সুয়াল ইভেন্টও স্পোর তৈরি হয়। এক্সাম্পল অফ দিস স্পোর্স আর অ্যাসকোব্যাসিডিয়াম জাইগো। Clear?
Yes, sir.
তো দিস থ্রি ইভেন্টস একটা সাইকেল করে দেখাই, ফাঙ্গাল লাইফ সাইকেল, সেক্সুয়াল লাইফ সাইকেল, পুরো তোমার কম্পাইল হয়ে যাবে। Sexual cycle, haploid mycelium organism, that is mycelium. They can produce gametes. Male gamete, আচ্ছা এরকম দেখাই বেটার। এখানে আমি আইসোগ্যামেটস দেখিয়েছি। Any can be possible, an iso or two gamete. Followed by these gametes are fused. তার মানে আমি কি শো করলাম? প্লাজমোগ্যামিটাইট, প্ল্যানোগ্যামেটিক অপুলেশন। Two N are fused. একটা কালার চেঞ্জ করতে হবে, ঠিক আছে, একটা ব্লু একটা ব্ল্যাক। Then fusion of nucleus forms zygote 2N. From this zygote, meiosis occurs. First plasmogamy, gamete formation, karyogamy, then meiosis. Event 1, 2, 3. 1, 2, 3. Meiosis produced 4 haploid spore. Check কর haploid spores. Each of these spores now grow, germinate, and form haploid further organism. Germination done by mitosis or meiosis? Germination
Sir,
মানে সেলে গ্রোথ হচ্ছে। হ্যাঁ,
Sir,
mitosis.
mitosis.
Mitosis. So germinate means here it create mitosis. So chromosome number is same, ploidy is same, N to N. An organism develop mycelium hyperstructure, complex hyperstructure mycelium develop. That means germination হয়ে আবার further cycling করবে। Sexual cycle of fungal sexual cycle: plasmogamy, karyogamy, meiosis, production of haploid meiospore, germination, haploid organism.